Hello, my name is Alex Pellegrini, and I'm going to explain to you the stages of mitosis. So, mitosis begins with interphase, and then after interphase, there is prophase. Prophase is the first stage of mitosis, the stage where the cell spends approximately 80% of its time. At the beginning of prophase, the membrane that surrounds the nucleus disintegrates, which exposes the chromatin. So there it is disintegrating, and in there those colors are the chromatin. The Golgi and endoplasmic reticulum are dispersed. The nuclear envelope breaks down, and the spindle apparatus, the cytoskeleton, is disassembled, which allows for the mitotic spindle to form. The mitotic spindle, or spindle apparatus, is composed of microtubules that form in order to ultimately connect the opposing poles of the cell. So these yellow right here is the spindle apparatus. The chromosomes begin to condense with the nucleus, and the sister chromatids are linked by the centromere, which serves as the binding site for the spindle microtubules. So these are the chromosomes in there, the same thing as the chromatids, the blue and the red. So mitosis only spends 20% of its time in the next 4 stages. So prometaphase. Prometaphase is the second phase of mitosis. This is the process that separates the duplicated genetic material that is in the nucleus of the parent cell into 2 identical daughter cells. So separating the duplicated genetic material right here, separating, and the nuclear envelope breaks down. Chromosomes attach to the microtubules at kinetochores, and kinetochores are a protein complex which forms the centromeres for spindle apparatus attachment. So these are the chromosomes attaching to the yellow spindle apparatus, and spindle apparatus is the yellow. The sister chromatids are attached at the kinetochores of microtubules from opposite poles of the cell. So one pole, other pole, and they attach to the chromatids. The chromosomes move to the equator of the cell, so they start to move into the equator of the cell, as shown in the next phase when they are at the equator of the cell. So the next stage is metaphase. Metaphase is the third stage in mitosis. So during the third stage of mitosis, metaphase, the chromosomes become attached to the spindle fibers. Nuclear membrane is no longer present, and the chromosomes are aligned at the equator of the cell, also known as the metaphase plate. In this phase, the chromosomes are attached to opposite poles, attached to pole here and pole here, by the spindle apparatus, and are under tension, so being pulled by the spindle fibers. So then, in the fourth phase of mitosis, anaphase, the chromosomes move away from one another. So as you can see, the spindle apparatus fibers are pulling the chromosomes apart to the poles. The proteins holding centromeres of sister chromatids are degraded, which frees the individual chromosomes. So the centromeres are degraded. The centromeres are like in the middle right there. So the spindle poles move apart, and the chromosomes are pulled to opposite poles. Then, in the final stage of mitosis, telophase, this is the final stage of cell division, mitosis. During this stage, the chromatids or chromosomes move to opposite ends of the cell, and 2 nuclei are formed. During telophase, the nuclear envelope reforms. So as you can see, the nuclear envelope in the dark blue, almost black, is reforming. and the Golgi and the endoplasmic reticulum reform as well. The chromosomes are clustered at opposite poles in the spindle apparatus. Oh my god. And the spindle apparatuses the spindle microtubules have retracted, and the cell is ready to split apart. And that is mitosis. Right there. So you can see. Thank you.